Welcome everyone to the video lecture for Unit 3, Module 6.2. In this one, we're going to talk more about specific and non-specific defenses, which we highlighted at the end of the previous module. As always, here are your learning objectives. Be sure to reference these when studying for your lecture quiz as well as your unit exam. Let's go ahead and get started. So there are 7 major categories of innate or non-specific immunity. We have 1, physical barriers, 2, phagocytes, 3, immune surveillance, 4, interferons, 5, the complement, 6, inflammatory response, and 7, fever. Innate or nonspecific defenses prevent the approach, deny the entry, or limit the spread of microorganisms or other environmental hazards. The figures that are on this slide and the next give a brief overview of the 7 types. But to cause trouble, an antigenic substance or a pathogen must enter body tissues. In other words, it must cross an epithelium, either at the skin or across a mucous membrane. The epithelial covering of the skin has multiple layers, a keratin coating, and a network of desmosomes that lock adjacent cells together. These barriers are effective in protecting underlying tissues. Even alongside the more delicate internal passageways of the respiratory, digestive, and urinary and reproductive tracts, epithelial tissues are tied together by tight junctions. These cells generally are supported by a dense and fibrous basement membrane. In addition, specialized accessory structures and secretions protect most epithelia. The hairs on most areas of your body provide some protection against mechanical abrasions, especially on the scalp. They often prevent hazardous materials or insects from contacting your skin. The epidermal surface also receives the secretions of sebaceous and sweat glands. These secretions flush the surface and wash away microorganisms and chemicals. Such secretions may also contain chemicals that kill bacteria, destructive enzymes called lysosomes, and antibodies. The epithelial lining of the digestive, respiratory, urinary, and reproductive tracts are more delicate, but they are equally well defended. Mucus bathes most of the surfaces in your digestive tract. Your stomach contains a powerful acid that can destroy many pathogens, and mucus moves across the respiratory tract lining. Urine flushes the urinary passages, and glandular secretions do the same for the reproductive tract. Special enzymes, antibodies, and acidic pH add to the effectiveness of these secretions. Phagocytes serve as janitors and police in peripheral tissues. They remove cellular debris and respond to invasion by foreign substances or pathogens. Phagocytes are the first line of cellular defense against pathogenic invasion. Many phagocytes attack and remove microorganisms even before lymphocytes detect them. The human body has 2 general classes of phagocytotic cells: microphages and macrophages. Microphages are the neutrophils and the eosinophils that normally circulate in the blood. These phagocytotic cells leave the bloodstream and enter peripheral tissues that have been subjected to injury or infection. Eosinophils are less abundant. They target foreign substances or pathogens that have been coated with antibodies. Macrophages are large, actively phagocytic cells. Your body contains several types of macrophages. Most are derived from the monocytes of the circulating blood. Typically, macrophages are either fixed in position or freely mobile. As a result, they are usually classified as fixed macrophages or free macrophages, but the distinction is not generally absolute. During an infection, fixed macrophages may lose their attachments and begin roaming around the damaged tissue. No organs or tissues are dominated by phagocytes, but almost every tissue in the body shelters resident or visiting macrophages. This relatively diffuse collection of phagocytes has been called the monocyte macrophage system, or the reticuloendothelial system. So how does an activated macrophage respond to a pathogen? It may engulf a pathogen or other foreign object and destroy it with lysosomal enzymes, bind to remove a pathogen from the interstitial fluid, but be unable to destroy it by the invader until assisted by other cells, destroy its target by releasing toxic chemicals such as tumor necrosis factor, nitrous oxide, or hydrogen peroxide into the interstitial fluid. And we'll consider these responses further in a later section. Fixed macrophages, or histiocytes, reside in specific tissues and organs. These cells are normally incapable of movement, so their targets must diffuse or otherwise move through the surrounding tissues until they are within range. Fixed macrophages are scattered among the connective tissues, usually in close association with collagen or reticular fibers. They're found in the papillary and reticular layers of the dermis, in the subarachnoid space of the meninges, and in bone marrow. In some organs, the fixed macrophages have special names. Microglia are macrophages in the central nervous system, and the Kupffer cells are macrophages located in and around the liver sinusoids. Free macrophages, or wandering macrophages, travel throughout the body. They arrive at the site of an injury by migrating through adjacent tissues or by leaving the circulating blood. Some tissues contain free macrophages. For example, alveolar macrophages, also known as phagocytic dust cells, monitor the exchange surfaces of the lungs for respiratory pathogens. Free macrophages and macrophages function in similar ways. Both can move through capillary walls by squeezing between adjacent endothelial cells. This process is known as immigration, or diapedesis. The endothelial cells in an injured area develop membrane markers that signal passing blood cells that something is wrong. The phagocytic cells then attach to the endothelial lining and migrate through the surrounding tissues. Both may be attracted to or repelled by chemicals in the surrounding fluids, a phenomenon called chemotaxis. They are particularly sensitive to cytokines released by other body cells and to chemicals released by pathogens. For both, phagocytosis begins with adhesion, the attachment of the phagocyte to its target. In adhesion, receptors on the plasma membrane of the phagocyte bind to the surface of the target. Adhesion is followed by the formation of a vesicle containing the bound target. The contents of the vesicle are digested once the vesicle fuses with lysosomes or peroxisomes. All phagocytic cells function in much the same way, although their targets may differ. The lifespan of an actively phagocytic cell can be rather brief. For example, most neutrophils die before they've engulfed more than 25 bacteria, and during an infection, a neutrophil may attack that many in an hour. The immune system generally ignores the body's own cells unless they become abnormal in some way. Natural killer cells are lymphocytes responsible for recognizing and destroying abnormal cells when they appear in peripheral tissues. The constant monitoring of normal tissues by NK cells is called immune surveillance. The plasma membrane of an abnormal cell generally contains antigens that are not found on the membranes of normal cells. NK cells recognize an abnormal cell by detecting those antigens. NK cells are much less selective about their targets than are other lymphocytes. They respond to a variety of abnormal antigens that may appear anywhere on a plasma membrane. They also attack any membrane containing abnormal antigens. As a result, NK cells are highly versatile. A single NK cell can attack bacteria in the interstitial fluid, body cells infected with viruses, or cancer cells. NK cells respond much more rapidly than T cells or B cells. The activation of T cells and B cells involves a complex, time-consuming sequence of events. NK cells respond immediately on contact with the abnormal cells. So let's talk about some of the process of NK activation here. 1. Recognition and adhesion if a cell has unusual components in its plasma membrane. An NK cell recognizes that that cell is abnormal. The first step is recognition and adhesion. If a cell has unusual components in its plasma membrane, an NK cell recognizes that cell as abnormal. The recognition activates the NK cell, which then adheres to its target. 2 is the realignment of the Golgi apparatus. The Golgi apparatus moves around the nucleus until the maturing face points directly towards the abnormal cells. The process might be compared to the rotation of a tank turret to the point a cannon at an enemy. The Golgi apparatus then produces a flood of secretory vesicles that contain proteins called perforins. These vesicles move through the cytosol towards the cell surface. 3 is the secretion of porphyrin. These porphyrins are released at the NK cell surface by exocytosis and diffuse across the narrow gap between the NK cell and its target. 4 is the lysis of the abnormal cell. On reaching the opposing plasma membrane, porphyrin molecules interact with one another and with the membrane to create a network of pores in it. These pores are large enough to allow free passage of ions, proteins, and other intercellular materials. As a result, the target cell can no longer maintain its internal environment, and it quickly disintegrates. So why doesn't porphyrin affect the plasma membrane of the NK cell? The answer is not really clear, but the NK cell membranes contain a second protein called protectin. It may bind and inactivate porphyrin. NK cells attack cancer cells and body cells infected with viruses. Cancer cells probably appear throughout life, but their plasma membranes generally contain unusual proteins called tumor-specific antigens, which NK cells recognize as abnormal. The NK cells then destroy the cancer cells, preserving tissue integrity. Unfortunately, some cancer cells avoid detection, perhaps because they lack tumor-specific antigens or because these antigens are covered in some way. Other cancer cells are able to destroy the NK cells that detect them. This process of avoiding detection or neutralizing body defenses is called immunological escape. Once immunological escape has occurred, cancer cells can multiply and spread without interference by NK cells. In viral infections, the virus replicates inside the cell, beyond the reach of the circulating antibodies. However, infected cells incorporate viral antigens into their plasma membrane, and NK cells recognize these infected cells as abnormal. By destroying them, NK cells can slow or prevent a viral infection. Interferons are small proteins released by activated lymphocytes and macrophages, and by tissue cells infected with viruses. An interferon binds to the surface of receptors on the membrane of a normal cell, and by second messengers, triggers the production of antiviral proteins in the cytoplasm. Antiviral proteins do not prevent viruses from entering the cell. Instead, they interfere with viral replication inside the cell. In addition to slowing the spread of viral infections, interferons stimulate the activities of macrophages and NK cells. At least 3 types of interferons exist: interferon alpha, beta, and gamma. Most cells, other than lymphocytes and macrophages, respond to viral infection by secreting interferon beta. Interferons are examples of cytokines, which are chemical messengers that tissue cells release to coordinate local activities. Most cells produce cytokines only for paracrine communication, that is, cell-to-cell communication within one tissue. However, defense cells release cytokines that also act as hormones, affecting the cells and tissues throughout the body. We'll discuss their role in regulating adaptive specific defenses a little bit later. Plasma contains over 30 special complement proteins that form the complement system. The term complement refers to the fact that the system complements the actions of antibodies. The complement proteins interact with one another in chain reactions or in cascades, reminiscent of those of the clotting system. The classical pathway activates the complement system most rapidly and effectively. It begins with the binding of complement protein C1 to nearby antibodies already attached to its specific antigen, such as bacterial cell walls. In the absence of antibody molecules, the alternative pathway, or the properdin pathway, activates the complement system. The alternative pathway is slower and less effective than the classical pathway. It begins when several complement proteins, including properdin or factor P, factor B, and factor D, interact in the plasma. Exposure to foreign materials such as the cell wall of a bacterium can trigger this interaction. Like the classical pathway, the alternative pathway ends with the conversion of an active C3 protein to the activated C3b protein. Complement activation brings about the following effects: pore formation, enhanced phagocytosis, and histamine release. Inflammation, or the inflammatory response, is a localized tissue response to injury. Inflammation produces local redness, or rubor, swelling, which is tumor, heat, which is calor, and pain, which is dolor. These are known as the so-called cardinal signs and symptoms of inflammation. Sometimes a 5th symptom is also included, which is just loss of function. Many stimuli can produce inflammation. They include impact, abrasion, distortion, chemical irritation, infection by pathogens, and extreme temperatures, or hot or cold. Each of these stimuli kills cells, damages connective tissue fibers, or injures the tissue in some other way. The changes alter the chemical composition of the interstitial fluid. Damaged cells release prostaglandins, proteins, and potassium ions. The injury itself may have introduced foreign proteins or pathogens, and these changes in the interstitial environment trigger the complex process of inflammation. Inflammation has several effects. The injury is temporarily repaired, and additional pathogens are prevented from entering the wound. The spread of the pathogens away from the injury is slowed. Local, regional, and systemic defenses are mobilized to overcome the pathogens and facilitate permanent repairs, and this repair process is called regeneration. Mast cells play a pivotal role in the inflammatory response. Comparable events that take place in almost every tissue subjugated to physical damage or infection. When stimulated by mechanical stress or chemical changes in the local environment, mast cells release histamine, heparin, prostaglandins, and other chemicals into the interstitial fluid. The histamine makes capillaries more permeable and speeds up blood flow to the area. The combination of abnormal tissue conditions and chemicals released by mast cells stimulates local sensory neurons, producing sensations of pain. The person may then take steps to limit the damage, such as removing a splinter or cleaning a wound. The increased blood flow reddens the area and raises the local temperature. These changes increase the rate of enzymatic reactions and accelerate the activity of phagocytes. The rise in temperature may also denature foreign proteins or vital enzymes of invading microorganisms. Because vessel permeability is increased, clotting factors and complement proteins can leave the bloodstream and enter the injured or infected area. Clotting does not take place at the actual site of injury due to the presence of heparin. However, a clot soon forms around this damaged area, both isolating the region and slowing the spread of chemical or pathogen into healthy tissues. Meanwhile, complement activation through the alternative pathway breaks down the bacterial cell walls and attracts phagocytes. Neutrophils are also drawn to the area by chemotaxis, where they attack debris and bacteria. As neutrophils circulate through a blood vessel in an injured area, they undergo activation. In this process, they 1. Stick to the side of the vessel and move into the tissue by diapedesis. 2. Their metabolic rate goes up dramatically, and while this respiratory burst continues, they generate reactive compounds such as nitric oxide and hydrogen peroxide that can destroy engulfed pathogens. And 3, they secrete cytokines that attract other neutrophils and macrophages to the area. As inflammation proceeds, the foreign proteins, toxins, microorganisms, and active phagocytes in the area activate the body's specific defenses. Fixed and free macrophages engulf pathogens and cell debris. At first, neutrophils outnumber these cells. Then, as the macrophages and neutrophils continue to secrete cytokines, the number of macrophages increases rapidly. Eosinophils may get involved if antibodies coat the foreign materials. The cytokines released by active phagocytes stimulates fibroblasts in the area. The fibroblasts then begin forming scar tissue that reinforces the clot and slows the invasion of adjacent tissues. Over time, the clot is broken down and the injured tissues are either repaired or replaced by scar tissue. The process is essentially complete, although subsequent remodeling may take place over a period of years. After an injury, tissue conditions generally become even more abnormal before they begin to improve. The tissue destruction that occurs after cells have been injured or destroyed is called necrosis. The destruction begins several hours after the initial event, and the damage is due to lysosomal enzymes. Lysosomes break down by autolysis, releasing digestive enzymes that first destroy the injured cells and then attack surrounding tissues. As local inflammation continues, debris, fluid, dead and dying cells, and necrotic tissue components accumulate at the injury site. This viscous fluid mixture is known as pus. An accumulation of pus is an enclosed tissue space called an abscess. Fever is a body temperature greater than 32.7 degrees Celsius or 99 degrees Fahrenheit. Circulating proteins called pyrogens can reset this thermostat and raise the body temperature. A variety of stimuli either act as pyrogens themselves or stimulate macrophages to release pyrogens. These stimuli include pathogens, bacterial toxins, and antigen-antibody complexes. Active macrophages release a cytokine called tumor necrosis factor, or TNF, which directly causes fever or stimulates the release of endogenous pyrogen, or interleukin-1, which is also abbreviated as IL-1. Within limits, a fever can be beneficial. High body temperatures may inhibit some viruses and bacteria. The most likely beneficial effect is on body metabolism. For each 1 degree Celsius rise in body temperature, metabolic rate increases by 10%. Cells can move faster and enzymatic reactions take place more quickly. As a result, tissue defenses can be mobilized more rapidly and the repair process speeds up. Adaptive or specific defenses result from the coordinated activities of T cells and B cells. These cells respond to the presence of specific antigens. In general, T cells bring about cell-mediated immunity or cellular immunity, which defends against abnormal cells and pathogens inside cells. B cells provide more antibody-mediated immunity or humoral immunity, which defends against antigens and pathogens in body fluid. Both kinds of immunity are important because they come into play and under different circumstances. Activated T cells do not respond to antigens in solution, and antibodies produced by activated B cells cannot cross plasma membranes. Moreover, helper T cells play a crucial role in antibody-mediated immunity by stimulating the activity of B cells. We can classify immunity as either innate or adapted. As we stated earlier, innate immunity is genetically determined. It is present at birth and is not related to previous exposures to a particular antigen. For example, people do not get the same diseases that goldfish do. Innate immunity breaks down only in the case of AIDS or other conditions that depress all aspects of specific resistance. Adaptive immunity is not present at birth. Instead, you have to develop immunity to a specific antigen only when you've been exposed to that antigen. Adaptive immunity can be active or passive, and these forms of immunity can be either naturally acquired or artificially induced. Active immunity develops after exposure to an antigen. It's a consequence of the immune response. In active immunity, the body responds to an antigen by making its own antibody. The immune system is capable of defending against a huge number of antigens. However, the appropriate defenses are mounted only after you encounter a particular antigen. Active immunity can result from natural exposure to an antigen in the environment, or naturally acquired from active immunity, or from deliberate exposure to an antigen, artificially induced active immunity. Naturally acquired active immunity normally begins to develop after birth. It continues to build as you encounter new pathogens or other antigens. You might compare this process to development of a child's vocabulary. The child begins with a few basic common words and learns new ones as they are encountered. Artificially induced active immunity stimulates the body to produce antibodies under controlled conditions so that you'll be able to overcome natural exposure to pathogens in the future. This is the basic principle behind immunization or vaccination to prevent disease. A vaccine is a preparation designed to induce an immune response. It contains either a dead or inactive pathogen or antigens derived from that pathogen. There are other types of vaccines now. As we know, the COVID vaccine is an mRNA vaccine, but we're not going to get too deep into that. Passive immunity is produced by transferring antibodies from one source to another. In naturally acquired passive immunity, a baby receives antibodies from the mother either during gestation by crossing the placenta or in early infancy through breast milk. It's also one of the reasons that typically we prefer to do a vaginal or true birth against a cesarean section, because the baby can acquire some of these things as they move through the vaginal tract. In artificially induced passive immunity, a person receives antibodies to fight infection or prevent diseases. For example, someone who's been bitten by a rabid animal gets injections containing antibodies against the rabies virus. Regardless of the form, adaptive immunity has 4 general properties. 1, specificity, 2, versatility, 3, memory, and 4, tolerance. A specific defense is activated by a specific antigen, and their immune response targets that particular antigen and no others. Specificity results from the activation of appropriate lymphocytes and the production of antibodies within targeted effects. Specificity occurs because T cells and B cells respond to the molecular structure of an antigen. The shape and size of each antigen determine which lymphocytes will respond to it. Each T cell or B cell has receptors that will bind to one specific antigen, ignoring all others. The response of an activated T cell or B cell is equally specific. Either lymphocyte will destroy or inactivate that antigen without affecting other antigens or normal tissues. Millions of antigens in the environment can pose a threat to health. Over a normal lifetime, you encounter only a fraction of that number, perhaps tens of thousands of antigens. Your immune system, however, has no way of anticipating which antigens it will encounter. It must be ready to confront any antigen at any time. Versatility results in part from the large diversity of lymphocytes present in the body, and in part from variability in the structures of synthesized antibodies. During development, cells in the lymphatic system differentiate to produce a huge number of lymphocytes with varied antigen sensitivities. The trillion or more T cells and B cells in your body include millions of different lymphocyte populations distributed throughout the body. Each population contains several thousand cells with receptors in their membranes that differ from the receptors of other lymphocyte populations. As a result, each population of lymphocyte responds to a different antigen. Several thousand lymphocytes are not enough to overcome a pathogenic invasion. However, when activated by an appropriate antigen, a lymphocyte begins to divide, producing more lymphocytes with the same specificity. All the cells produced by these divisions make up a clone, and all members of that clone are sensitive to the same specific antigen. To understand how the system works, think about running a commercial kitchen with only samples on display. You can take a display a wide selection because the samples don't take up much space, and you don't have to expand energy now preparing food that might never be eaten. When a customer selects one of your samples and places an order for several dozen, you can prepare them on the spot. The same principle applies to the lymphatic system. Your body contains a small number of different kinds of lymphocytes. When an antigen arrives, lymphocytes sensitive to it are selected. These lymphocytes divide to generate a large number of additional lymphocytes of the same type. As we just saw, during the initial response to an antigen, lymphocytes that are sensitive to it undergo repeated cycles of cell division. Immunologic memory exists because those cell divisions produce 2 groups of cells. One group attacks the invader immediately. Another group remains inactive unless it meets the same antigen at a later date. This inactive group is made up of memory cells that enable your immune system to remember an antigen it has previously encountered, and to launch a faster, stronger, and longer-lasting counterattack if such an antigen appears again. The immune system does not respond to all antigens. All cells and tissues in the body, for example, contain antigens that normally do not stimulate an immune response. We say that the immune response exhibits tolerance towards those antigens. The immune response targets foreign cells and compounds, but it generally ignores normal tissues. During their differentiation in the red bone marrow, B cells and thymus T cells, any cells that react to antigens that are normally present in the body are destroyed. As a result, mature B cells and T cells ignore normal antigens, also called self antigens, but attack foreign antigens or non self antigens. Tolerance can also develop over time in response to chronic exposure to an antigen in the environment. Such tolerance lasts only as long as the exposure continues. When an antigen triggers an immune response, it generally or usually activates both T cells and B cells. The activation of T cells generally occurs first, but only after phagocytes have been exposed to the antigen. Once activated, T cells attack the antigen and stimulate the activation of B cells. Activated B cells mature into cells that produce antibodies. Antibodies in the bloodstream then bind and attack to the antigen, and we'll examine this process a little bit more in the next module.